Hello. So, very good evening to the student who have joined. Can you please let me know in the chat box if my voice is audible and if you can see my video and also the PPT. Okay, thank you so much. So, we will wait till other students join, then we will start today's session. So very good evening to the students who have joined. We will wait for 2 to 3 more minutes and then we will start today's session. Meanwhile, if you have any problem for seeing the PPT or listening to my voice, please let me know in the chat box. Let us wait till it is 6 p.m. dot and then let's start the session. Again, I am saying if you have any problem for listening to my voice or seeing the PPT, please let me know right now. And if everything is alright, then it's fine. Okay, I think we can start with the introductions. Okay, so very good evening to the students who have joined. My name is Mousumi Deb and I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am the TA for your course NPTEL course of Organic Chemistry in Biology and Drug Development. Today it is our week 4 live session, and in this session we will discuss the week 5 course content. Right now the week 5 is live. So today we will discuss the content, the course content, the topics which are covered in week 5. So like every week, today also we will discuss around 20 questions regarding your week 5 content, and all the questions will be MCQ form. So today also we will discuss around 20 questions. All the questions, it will be in MCQ form. So these type of question will help you for understanding the course more better, and also they will help you for your weekly assignment. Okay, if you practice this kind of question, it will help you to do your assignment which is given to you in this week 5 also. So before starting, as I always ask if my voice is audible and the presentation is clear. If you have any problem, please let me know in the chat box. So for the smooth running of the class, I have just muted off all of you, but you can just always communicate with me through the chat box. Okay, the chat box is open and I am always monitoring the chat box. So if you have any doubt, any query or anything you want to say me, you can just type it in the chat box. Okay, once I show you the questions, I want all of you to try to answer the questions, okay, in the chat box. It does not matter if it is correct or wrong, because no marks will be given for the class also. But if you try to answer the question, and if you answer the question, it will be better for you only, because it will help you to know that how much you have understood the course so far. So before starting, I will just sum up a few things, just like every week. So there will be a proper problem solving session every week. The session will be on Tuesday from 6 to 8 p.m. Sessions will be live on Google Meet platform. If any network disruption happens, please wait for a few minutes. Do not leave the session. I will try to join as soon as possible. Okay, so this I always tell that if any kind of network disruption happens, it may happen at your side also, it may happen at my side also. So please do not leave the class if it happens at my side, because I will try to join as soon as possible. Okay, and during the session students can post their question or doubts through the chat box. Just I have said it 2 minutes earlier that if you have anything you want to tell me regarding the questions, regarding the session, if you have any problem listening my voice or everything, just let me know in the chat box. And if still the doubt persists after class, the students can drop an email to me, because I will answer those queries through email. I will share my email ID at the end of the session, and you can just drop an email to me. I will help you through the email also. So this presentation and class recording will be shared to you very soon from the NPTEL side. Okay, so the presentation which I am showing to you today, this PPT, and also the recording of today's class, all of it will be shared to you, okay, in due course of time. So you don't have to worry if any network problem happens at your side. So I think it is clear. So I will just start today's session. Maybe when students will join, eventually I will add them. So so far my voice is clear, and you can also see the chat box, right? Sorry, I mean you can see the presentation. So everything is fine, right? Let me know in the chat box. Yeah, so thank you so much. I think we can start the session, and if other students, when they will join, I will eventually let them in. Okay, so today is the week 4 live session. So like we are going ahead in every week. So in this week 4 live session, what we will do is we will discuss week 5 course content. Now what it is in the year week 5 of this course, the topics which are covered in week 5 are synthetic biology. So we have discussed synthetic biology in last week also. So today it will be continued for few questions. Then we will move forward to the nucleic acid. After nucleic acid, we will go to the DNA sequencing method. So these are the topics for today's session and to this week 5. So I think I can start the session. So as I have always said, I will show you a question and the 4 options will be given. You have to try to answer which of the following is the most appropriate answer. Okay, does not matter if it is right or wrong, but every student present in the class should try to answer this. Write the option you think is correct in the chat box, and then I will, after some time, I will show you the correct answer, and then we will discuss that topic. Okay, so let us start. The first question of today's session is, correct composition of nucleotides is option A, base plus phosphate, option B, base plus sugar, option C, sugar plus phosphate, and option D, base plus sugar plus phosphate. So you have to identify which is the correct composition for the nucleotide. Among A, B, C and D, you have to find out which of the following is the correct option. You can write your answer in the chat box. I will wait for a few minutes, then I will show you what is the correct answer. Then we will discuss in detail what is a nucleotide, what are the units of the nucleotide and everything. So what is the correct answer? Everybody try to answer it in the chat box. It is a basic question regarding your nucleotide topic. Okay, it's because your nucleic acid, when you start the nucleic acid, nucleotides are the basics of the formation units for the your nucleic acids, the DNAs and RNAs. So what is the correct composition of nucleotides? Base plus phosphate, base plus sugar, sugar plus phosphate, or base plus sugar plus phosphate. Let me check. Okay, option D is the most frequently given answer. Now let me check what is the correct answer and then let us discuss it. Okay. So the correct composition of nucleotides is, very good, it is number D, base plus sugar plus phosphate. Now there is 3 parts of a nucleotide. So this is a basic structure of a nucleotide. As you can see, there is a sugar in between, in the center of the structure. Okay, this is a 5-member ring. Okay, and this is called a pentose sugar. Okay, and now we have a base here. This is a nitrogenous base, which is a very important part for your nucleic acid. So there is a nitrogenous base, which is a very important part for your nucleotide, and also we have a phosphate unit. So basically these 3 units comprise of a nucleotide. Now as you can see, this is a particular structure. So various units are named differently. So as you can see, there is a base here. Okay, at the 1 position of this, if this is a sugar, which is a pentose sugar, this is a 1, this is 2, this is 3, and this is 5 position. Okay, sorry, this is the 4 position. So in the, and this is a 5, now 5th carbon. Okay, at the 1 position there is a base present like this. This nitrogenous base, it is bonded with this pentose sugar with a glycosidic bond. So this nitrogen and this carbon bond, it is called the glycosidic bond. Okay. Now if in this position, in this sugar position, if at 2 prime position, means at this position, if we have a OH group, okay, then it is called the ribose sugar. But if we have a hydrogen here, it is called the deoxyribose unit. Okay. Now if you have only base and you have only this sugar, it is called a nucleoside. So this part only base and this sugar unit, at they are, these two together are called nucleoside. Okay, and again what I was saying, yeah, the base and this sugar, they together we are called the nucleoside. If there is a 1-phosphate unit, then it is called the nucleoside monophosphate. If there is a 2-phosphate unit, it is called a nucleoside diphosphate. And if there is a 3rd phosphate unit, it is called the nucleoside triphosphate. Okay. So what are the type of bases? So basically 2 type of bases are present in the nucleic acid. The first one is a purine kind of base. So what is the structure of purine? This is the structure of purine. So basically the purine is a bicyclic nitrogenous heterocycle. Okay, one is the 6-member ring, and there is a 5-member ring which is fused with the 6-member ring. And if you start numbering, this nitrogen is number 1, then it is 2, now this is 3, this is 4, 5, 6. After completing it, we have to just come here, it is 7, it is 8, and it is 9. And in this 9 position, the R group may be present. Okay, and this, with this 9th position, this is bonded with this glycoside bond. So this one, it is called the adenine base. We all know about the A, G, T, C bases which are present in DNA and RNAs. Okay, so now what is the full form of this bases? A stands for adenine, this is the structure of adenine. G stands for guanine, this is the structure of guanine. C stands for cytosine. This adenine and guanine, these are purine bases. And guanine, cytosine, uracil, thymine, these are pyrimidine bases. Now what is the structure of pyrimidine? Pyrimidine is a nitrogenous heterocycle which is 6-member in ring, and nitrogen are present in 1 and 3 number position. So this 1 position there is a nitrogen, 2 there is a carbon, 3 again there is a nitrogen, then 4, 5, 6 all are carbons. Here is also same, here it is also same. So cytosine, uracil and thymine, these 3 are pyrimidine bases, and you can see the structural difference between uracil and thymine. We all know in DNA thymine is present, while in RNA the uracil is present. Okay, and the if you can look at carefully at the structure of the your thymine and uracil, the only difference is at the 5th position there is a hydrogen in uracil, but there is a methyl in the thymine. Okay, so these are the purine and pyrimidine bases for the DNAs and RNAs. So this was a very basic question regarding your nucleotides and your nucleic acids. So I think now we can move forward to next question. Again I am saying, if you have any doubt or any query, you just let me know in the chat box. Okay, I have just muted the video call so that there is no disturbance, or there is the class is the smooth streaming is there, but the chat box is always open and I am checking the chat box always. So if you have any doubt regarding any question, any problem, just you can put it in the chat box. Okay, and if when I am telling you one question, please try to answer this question in the chat box. So for this question, more or less everybody has participated and everybody has given the correct answer. So now let us move toward the next problem. So problem number 2, it says that the sister strands of DNA molecule are, option number A, parallel and complementary, option B, anti-parallel and complementary, option C, parallel and identical, option D, none of these. So we all know in the DNA molecule there are two strands of DNA molecule, okay, which are forming the helix structure, and between these two strands there is a hydrogen bonding, right. So you have to identify, now you have to identify which of the following is the correct answer regarding that sister strands of the DNA. So the first option is these strands are parallel and complementary. Option B, these sister strands of DNA molecule are anti-parallel and complementary. Option C, these strands are parallel and identical. And option D, none of these. Now try to answer this. Which of the following you think is the correct one? Everybody should try to answer this question. Does not matter if your answer is wrong, but I think everybody should try. So is there any answer for this question? Okay, option B. So option B is the most frequently given answer, right? Okay, let me check what is the correct answer and then let us discuss. So the correct answer for this problem number 2 is, very good, it is number B, anti-parallel and complementary. Now let us discuss. So we all know that in the DNA we have a double helix structure, right. So one strand of a DNA is intertwined with other strand and it is called a double helix kind of structure. So we all know about it, right, like if one strand goes like this, there will be another strand of a DNA and it forms this kind of structure, okay, which is called the double helix. Now these strands are called the sister strands of a DNA molecule. Okay, now what is the basic features for these sister strands of the molecule? First, they will be anti-parallel. What is the mean by do we mean by anti-parallel? If one strand goes from the 5 prime to 3 prime end, another strand it will go from the 3 prime to 5 prime end. Now the 5 prime end of a one strand is always parallel to the 3 prime end of the other strand. Okay, and the structure shows a complementary base pairing. So what is this 5 prime and 3 prime mean? If you look at carefully in the previous question, here you will see this is the 5 prime site, okay, this 5 prime strand and this is the 3 prime strand and this is the 5 prime. In the 5 prime region we have a phosphate unit, okay, and here if you look at carefully, you will see one phosphate unit which is in the 5 prime of a particular ribose sugar. It is joined with the 3 prime hydroxyl group, okay, and this is called the phosphodiester bond, Okay, and if the one strand it goes to the 5 prime to 3 prime like this, another strand it is just opposite, it goes to the 3 prime to 5 prime end. So that's why these are anti-parallel. Okay, and complementary in that sense, like if we have thymine in one side and we will have adenine in other side. So we all know like thymine bonds with adenine, right? In DNA we DNA and RNA we all know what happens in DNA. Adenine it bonds with thymine and guanine it bonds with your cytosine. Okay, so this is a purine kind of base, this is a pyrimidine kind of base. This is also a purine kind of base and this is a pyrimidine kind. So one purine and one pyrimidine they bond together. In between adenine and thymine there is a two hydrogen bonds are present. Okay, 2 hydrogen bonds are present there, and in guanine and cytosine 3 hydrogen bonds are present. If you look at this picture carefully, you will see here is 2 hydrogen bonds are present here, and here 3 hydrogen bonds are present. Now complementary means if one strand you have like thymine and then you have guanine and then we have cytosine, in another strand what we will have the other base pairs with which they are bonding, like thymine bonds with adenine, guanine bonds with cytosine, cytosine bonds with guanine. So this is the complementary strand. So the sister strands of DNA they will be anti-parallel and complementary. So I hope it is clear to all, and most of you have given the answer correctly. So I hope it is clear. Even if it is not clear, please let me know in the chat box. Okay, at the end of the session I will discuss it once more. The questions which you feel I need to discuss it once more, just note down the number, and at the end of the session just tell me, I will repeat this. So now we will move toward the next problem. So here it was, here the picture was here. Once again I will erase this part. So as I have said, adenine bonds with thymine and your guanine bonds with cytosine, and in adenine thymine there are 2 hydrogen bonds. If you look at the structure carefully, this is the structure of your adenine and this is the structure of your thymine, and that is how the hydrogen bonding happens. Okay, in guanine and cytosine this type of hydrogen bonding happens. So I think problem number 2 it is clear to all. So quickly I will move toward the next problem. So problem number 3 it says that which of the following is known as catalytic antibody. Your options are number A abzyme, option B harpen, option C epitope, and option D paratope. So which of the following is known as catalytic antibody? Your 4 options are like this: option A abzyme, option B harpen, option C epitope, and option D paratope. Now which of the following is correct? Let me know in the chat box. Everybody try. Even if it is wrong, it doesn't matter, but everybody should try it. Okay. So let me check in the chat box if there is any answer for this question. So I think I have a few answers for this. Number A, option B, option A, B. So option A and option B are most frequently given, right? Okay, let us check now and then let us discuss what is a catalytic antibody. So the correct option is number A, abzyme. Abzymes are known as catalytic antibody. Now what is abzymes? An abzyme is an antibody that expresses catalytic activity. A single molecule of an antibody enzyme or abzyme is capable of catalyzing the destruction of thousands of target molecules. An enzyme is used to lower the activation energy of the reaction, allowing for the transition state to be possible and the product to be formed. An enzyme are typically artificially made and is made by having the immune system make antibodies that bind to a molecule that resembles the transition state of a catalytic process. Now, production of enzymes, what are the methods through which the enzymes can be produced? Antibody molecules are produced by the immune system to bind and neutralize foreign substance called antigens. Foreign proteins of bacteria and viruses are called herpins. They act as antigens. Transition state analogues are molecules which are more stable than the transition state itself, but they mimic its 3D structure. If injected into the bloodstream of an animal, transition state analogues act as herpins and they elicit the antibody production. Abs are isolated from the serum of the animal and used as enzymes. The enzymes, they bind to the transition state molecule and it may be expected to catalyze the corresponding chemical reaction by forcing the substrates into a transition state geometry. So, that is how the production of enzymes happens. What are the examples of the enzymes? The hydrolysis of a hydroxy ester by enzymes. Hydroxy ester forms a cyclic intermediate during hydrolysis. Cyclic phosphate ester is the structural analog of the cyclic intermediate. This analog is used as an antigen to elicit the antibodies, and these antibodies bind the cyclic intermediate, increasing the reaction rate. So, it was an example of a enzymes. So, now we all know what is a enzymes, and these are known as catalytic antibody because they act as a antibody which expresses the catalytic activity. And how it is produced, we all have read it, like the antibody molecule, they are produced by immune system to bind the foreign substance, and when the transition state analogues of a molecule are which are more stable than transition state itself, they actually act as a mimic, and they, when these are injected into bloodstream, they acts as a herpin and antibody production happens, and that is how enzymes express their effects. Okay. So, now moving toward the problem number 4. So, problem number 4 states that the structure of DNA is closely related to its biological function. Which of the following statements regarding the structure of DNA is correct? Let me just okay. So, we was in the problem number 4. It says the structure of DNA is closely related to its biological function. Which of the following statements regarding the structure of DNA is correct? Number A, all base pairs interact by forming 3 hydrogen bonds. Number B, DNA is comprised of nucleotide subunits, each containing a base, a deoxyribose sugar and a phosphate group. Option C, the double helix of DNA is stabilized by interaction between adjacent ribose sugars. Option D, each DNA sequence folds into a unique and complex 3D structure. Now, identify which of the following statements are correct regarding the structure of DNA. Let me know in the chat box among A, B, C and D which of the following you think is the correct answer. Read the options very carefully. I know you can do it because it is a rather very basic question. If you read the options carefully, you can easily identify the correct option. Okay. let me check the chat box now. Option B, option B, option B. So option B and one option C. Okay, let us check. So yeah, very good. The correct answer is option B. DNA is comprised of nucleotide subunits, each containing a base, a deoxyribose sugar and a phosphate group. So there was a option C. So if you look at the option C carefully, it states that the double helix of DNA is stabilized by the interactions between adjacent ribose sugar. But in the previous I think first or second question, we have seen that the interaction between the sister strands of the DNA, they actually happens between the base pairs. Okay, the complementary base pairs, they form hydrogen bond among themselves, and after that the DNA double helix gets stabilized with that particular interaction. And all base pair, they do not interact by 3 hydrogen bonds. Adenine and thymine, they have 2 hydrogen bonds, and guanine and cytosine, they have 3 hydrogen bonds. So here there is another example of the chemical structure of the DNA. So the sugar phosphate backbone is there, okay, and with this backbone, DNA is a polymer which is made of the units called nucleotides. The nucleotides are made of 3 different components, a sugar, a phosphate group and a base. These are 4 different, there are 4 different bases: adenine, thymine, guanine and cytosine in case of the DNA, and in case of RNA we do not have this thymine, we will have uracil in that place. So what holds the DNA strands together? DNA strands are held together by the hydrogen bonds between the bases on adjacent strands. Adenine always pairs with thymine, while guanine always pair with cytosine. Adenine pairs with uracil in case of RNA. So there is adenine and thymine, we have 2 hydrogen bonds here. In between guanine and cytosine, we have 3 hydrogen bonds. From DNA to proteins, the bases on a single strand of DNA act as a code, and the letters from these 3 letter codes which code for amino acid and the building blocks of the protein. So DNA, after the they form RNA after the from transcription, and RNA they give rise to protein in a process called translation. Okay, an enzyme called RNA polymerase, they transcript DNA into mRNA messenger ribonucleic acid, and it splits apart the 2 strands from the double helix and then reads the strand and copies the sequence of the nucleotide. And the difference between RNA and original DNA is that in place of thymine we will have the uracil. So if it is a DNA sequence here like TT, CCT, GAA, CCC, GTTA, our mRNA sequence will be only that in place of T we will have a U, uracil, and other everything will be same. The mRNA sequence will be now UU, CCU, GAA, CCC, GUUA, and every 3 of this base are actually act as a code for a new amino acid, and that is how the protein is formed. So this RNA from the protein synthesis for the RNA, mRNA, tRNA, everything we will discuss in the later weeks. Okay, I think it is in your week 6 course content. It is not for this course content, but I just repeat it for since it was there here. So I think it is clear to all the problem number 4, and I think I can move toward the problem number 5. Again I am saying, if you have any doubt, any query, please do write in the chat box. Okay, and if you want me to repeat any question, just note down the number. At the end of the session I will repeat that question for you. Okay. So problem number 5, it is a simple mathematics. So it says that in a particular human DNA, it has been observed that adenine is present in the 27%. What will be the percentage of cytosine in that particular DNA sample? The options are given: option A 27%, option B 46%, option C 73%, and option D is 23%. Now try to solve this and solve this question. It is very easy. Just remember that adenine bonds with thymine, guanine bonds with cytosine, and then try to solve this, and tell me in the chat box which of the following you think is the correct option. Sorry. It's a very basic maths. Just take one more minute and I will then show you the answer and how to solve this kind of question. It is very easy actually, it's a very basic and very easy. Okay, let me check in the chat box if you have answered it. Okay, I have more than one answer. I think in this case, can each DNA sequence have different 3D structure? No, actually each DNA sequence cannot have different 3D structures. Basically the 3D structure is the same. It is the double helix structure. Okay, there is no different 3D structure for each DNA sequence. So if the whatever the sequence may be, the 3D structure will be basically the same. So for the next question we have option like number D, number C. So number A, C and D, these three are given already. So let me check which is the correct answer. Okay, correct answer is number D, 23%. Now how to solve this question? So the rule of base pairing, as I have always mentioned, DNA, it is cytosine and guanine all together, and for adenine and thymine. So in RNA, cytosine bonds with guanine but adenine bonds with uracil. So adenine makes hydrogen bond with thymine. Since 27% of adenine is present, the percentage of thymine will also be 27% because adenine is bonding with thymine. So both of them will be present at the same amount. So thus the remaining two bases, cytosine and guanine, consist of total 100 minus 27 plus 27. Okay, if total is 100% and 27% of adenine and 27% of thymine is present, so what will be the remaining percentage? It will be 100 minus 27 plus 27 means 100 minus 54 means 46%. Now cytosine and guanine, they make hydrogen bond with each other, right? So hence they are present in a equal amount. And if cytosine and guanine are present in a equal amount, so what will be the percentage of your cytosine? It will be 46 divided by 2 means 23. Okay. So this is a very basic simple question, and I think if this type of question is given to you and if any base is given to you, I think you can now solve this question, right? If you have any doubt for regarding this mathematical problem, let me know in the chat box. Adenine bonds with thymine, so the same amount of adenine and thymine will be present and the remaining will be for the guanine and cytosine. If 27% of adenine is present means 27% of thymine is also present, and so guanine and cytosine will consist of 46%, and since guanine and thymine both are present in a same equal amount, so what will be the amount of the cytosine? It will be 46 divided by 2 means 23. Now moving toward the next problem. Problem number 6, it says which of the following reaction is not catalyzed by catalytic antibodies? Your options are number A, 5-exotate cyclization for epoxy alcohol, option B, 6-endotate cyclization of epoxy alcohol, option C, Claisen rearrangement at low temperature, and option D, isomerization by sigmatropic reaction. So this is an example of different reactions which may be catalyzed by catalytic antibody. You have to find out which of the following is not catalyzed by catalytic antibody. So let me check in the chat box if we have any answer for it. I think we have number A, number D. Okay, let me check which is the correct answer for this question. So, the correct answer is number A, 5-exotate cyclization for epoxy alcohol. This reaction is not catalyzed by catalytic antibody. So, stages involved in producing the catalytic antibody, I have also mentioned it in the I think question number 3 or something. So, first you have to choose a target reaction, and then the chemistry we have to identify the design of transition state analog for the organic synthesis, Harpen as a protein conjugate, then immunology, the hybridoma technology is there, and these all are involved in a molecular biology. Then we have to identify the novel catalytic antibodies which will guide the protein chemistry, and the improved design of this transition state analog we have to do it. So, what is the endotate cyclization for the exostate cyclization for a epoxy alcohol? So, these are called a Baldwin's rule. So, Baldwin rules are basically a series of guidelines which are given for a closure of a ring closure, means from acyclic to cyclic molecule. So, we have this Baldwin rule. So, this is not actually, this is basically a chemistry question. So, Baldwin's classification, there is a number of atoms are newly formed cycle. Okay. So, 3 prefixes are there. It is there is a tet, there is a trig, and there is a dig. Okay. So, if there is the one from, it is a sp hybridization, it is the dig. If it is sp2, it is the trig, and if it is the sp3, it is the tet, the hybridization at the point of the closure ring. Okay. And the number of the newly formed cycle, the atoms of newly formed cycle is there. So, it is a hash minus A minus B. So, this is the 3 prefixes are there. Okay, like 5-exotate, 5 it actually 5 it identifies like the number of atoms of a newly cyclic form, means it will be a 5-member ring. A, it is exo means if the newly position of the bond formed the cleaved during the cyclization, if it is outside the newly formed bond, and if it is inside the newly formed, it is called endo, and B, it is the hybridization of the close. So, tet means there is a sp3 hybridization. So, in this Baldwin rule, actually there are many rules regarding which reactions are. So, there is a many rules, like not every reaction is actually allowed, there is some allowed and some are not allowed. So, if a reaction is not allowed by the Baldwin rule, we cannot that particular reaction cannot also be catalyzed by catalytic antibody, since the transition state analog will not be that much of stable, and if it is not as much stable, we cannot use it, we cannot catalyze the reaction by catalytic antibody. So, among these 4, the first one will not be catalyzed by catalytic antibody, okay, because it is not actually much more feasible. So, moving toward the next problem. Problem number 7, it says that the term hyperchromic shift indicates number A, decrease in the absorption of UV light by a solution of DNA, number B, increase in absorption of UV light by a solution of DNA, number C, increase in the shift of wavelength of UV light by a solution of DNA, and option D, decrease in the shift of the wavelength of UV light in the solution of DNA. So, which of the following you think is the correct justification for the term hyperchromic shift? Let me know in the chat box. The term hyperchromic shift, it indicates decrease in the absorption of UV light by a solution of DNA, increase in the absorption of UV light by the solution of DNA. Number C, increase in the shift of the wavelength of UV light by the solution of DNA, or number D, decrease in the shift of wavelength of UV light by a solution of DNA. Let me check in the chat box. Okay, number B, so what are the options? Number C, B, D, C, B, D. So, we have 3 options, number C, number B, and number D. So, everybody is saying different answers. So, let us check which is the correct answer. So, the correct answer for this question is number B, increase in the absorption of UV light by a solution of DNA. So, this is, so there are various shifts which are indicated. Okay, so what are the various shifts which are basically related for a UV spectroscopy? So this is a particular wavelength. This side wavelength is increasing. This side the intensity of absorption is indicated by the y axis. Okay, and this is the maxima for the absorbance. Okay, this is the maximum absorption. If this maxima it shifts toward the higher wavelength, if it is shift toward the higher wavelength, it is called the bathochromic or red shift. And if this maxima it is shifted toward the lower wavelength, it is called the hypsochromic or blue shift. If the intensity of this absorption is increasing, okay, then it is called the hyperchromic shift, and if it is decreasing, it is called the hypochromic shift. So increase of the absorption means this is the this side it is absorption is indicated. If this is increasing, it is called the hyperchromic shift. So hyperchromic shift due to the increase in the intensity or Emax value. This is the Emax value, okay, the maximum intensity, and due to the introduction of a oxochrome, the hyperchromic shift happens. For example, pyridine, this absorbs the highest absorption value at 257 nanometer is 2750, but 2-methyl pyridine the highest at 262 it is Emax is 3560. Hypochromic shift, inverse of the hyperchromic shift, means the decrease in the intensity. The introduction of any group that compounds which is going to alter the molecular pattern of a compound results in the hypochromic shift. For biphenyl absorption happens at 250 nanometer, have an intensity of 19,000, whereas 2-methyl biphenyl has an intensity of 10,250. This is the example of hypochromic shift. So basically what happens during, so if the maxima, so there is a intensity goes there, okay, this is the intensity and this is the wavelength. If the maxima shifts toward the higher wavelength, this is the maxima. If it shifts toward the higher wavelength, it is called the red shift or bathochromic shift. If the maxima it is shifted toward the lower wavelength, it is called the blue shift or hypsochromic shift. And if the intensity of the maxima it is increased, if it is the original, if the intensity is increases, it is called the hyperchromic shift, and if the intensity is lower, it is called the hypochromic shift. Okay, so this is all terms which are related to the UV light and UV absorptions. Okay. Now next problem. Problem number 8, it says that what will be the correct statement regarding the melting temperature of the DNA? Options are: number A, in presence of urea, Tm, or the melting temperature it is denoted by this T suffix m. So this Tm value of DNA increases. Option B, in presence of urea, Tm value of DNA does not change. Option C, higher concentration of sodium chloride increases the Tm value for DNA. Option D, higher concentration of sodium chloride decreases the Tm value for DNA. Now which of the following is the correct statement regarding the melting temperature? Try to answer this question. After your trial, I will show you what is the correct answer, what is the melting temperature of a DNA and how it can be altered. So what is the correct statement? Have anybody answered it? Okay, I have few answers, number C and number D. So I have 2 number C, 2 number D. Okay, let me check. So the correct answer is number C, higher concentration of sodium chloride increases the melting temperature or Tm for the DNA. So what is the melting temperature? The temperature at which half of the DNA strands are single stranded and half are double stranded. the melting temperature is characteristic of a DNA composition. Higher G plus C content DNA has a higher melting temperature due to more hydrogen bonds. Okay. Base composition calculation, how the calculation happens, the base composition Tm transition the melting temperature equal to the 59.9 plus 0.41 into percentage of Gc minus 600 per wavelength. Okay. So, other are accurate methods are also available. So, basically what is the melting temperature at which half of the DNA present is single stranded and half is double stranded. The more the G and C value content, the more hydrogen bonding and higher the melting temperature. So, how the melting temperature can be altered? So, this is the ratio of double stranded and single stranded DNA, okay, and this is the melting temperature at which the half of DNA is there is the ratio is 1:1. Okay, so melting temperature of DNA can be increased by more GC base pairs and fewer AT base pairs because it increases the number of hydrogen bonds. High salt concentration and longer DNA strands. So, if we have higher salt concentration of sodium chloride, the melting temperature for DNA it will be increased. Okay. So, there are other methods what more guanine and cytosine base pairs and longer DNA strands. These will also make the melting temperature increase. Okay. Now, problem number 9, it says which pyrimidine base is absent in case of DNA. Your options are uracil, cytosine, thymine and adenine. In the first slide I mentioned what are the pyridine and what is the purine and what is the pyrimidine bases are present in DNA and RNA. So, among the pyrimidine bases, one pyrimidine base is not present in the DNA, and I think all of you should answer this question because it is a very basic question. Let me check the chat box. Yeah, I think everybody has answered it correctly. Number A, option number A, it is correct answer. So, yeah, uracil is absent in case of DNA. So, as I have mentioned it previous also, I will also discuss it in anyone. So, this is the structure of pyrimidine. As you, it is very important for you while you are taking a course like this, or which is mix of organic chemistry and also biology. Remembering the names of heterocycles are very important. So, basically this is a pyrimidine ring. Okay. So, what is the nature of this nitrogenous heterocycle? That it is a 6 member and it has 2 nitrogen atom at 1 and 3 position. And there is also purine base which is a bicyclic heterocycline, 1 6 member, 1 5 member fused together. We have total 4 nitrogen, and what are the numbering? Numbering goes like this: 1 2 3 4 5 6 7 8 and 9, and in this 9 position we will have this glycoside bonding. Adenine and guanine are present in both DNA and RNA, but for pyrimidine case uracil is only present in RNA and thymine is only present in DNA. Cytosine is present in both DNA and RNA. Okay. And the structure of uracil and thymine, as I have also mentioned it before, only difference is here at 5th position we do not have a methyl group. Here thymine in case of we have a methyl group, but for uracil we have a hydrogen only in this place. Okay. Now, moving toward the problem number 10, it says that the complementary strand of a primary single stranded DNA is formed by option A DNA ligase, option B DNA primer, option C DNA polymerase and option D DNA helicase. So, which of the following enzyme is involved in forming the complementary strand of a primary single strand DNA? Complementary strand of the primary single stranded DNA is formed by DNA ligase, DNA primer, DNA polymerase or DNA helicase. Let me check in the chat box. Number C. So number C is the mostly given answer. We have one number B, but others are number C. Okay, let me check. So correct answer is yes, it is DNA polymerase. Now let us discuss what are other enzymes. So what is the DNA polymerase? So as you can see, this is a double stranded DNA. So when there is a the DNA polymer, I mean when the formation of another DNA strand is happens, what happen? There is a enzyme. So there are various enzymes which are involved here. So as in these colors they depict the hydrogen, nitrogenous base. Okay. So here we have various enzymes involved in the case of this DNA polymerization. So we have original template DNA. Okay. So before the replication, DNA replication, what we will have, this is the replication fork. Okay. So what happens first, this there is a leading strand of a DNA and there is a lagging strand. Okay. And there is a DNA polymerase enzyme which actually takes the lagging strand and add one by one the bases are added, and there is another sister strands is formed from the primary single stranded DNA. So there are other enzymes which are also involved here, like there is a helicase enzyme. So what does this helicase do? They actually breaks this hydrogen bonds here, so that the single stranded DNA strand is formed. Okay. And there are other enzymes which are also involved in this particular case. So there is a leading strand here which is formed like this, and the nitrogenous bases are there. These are free nucleotides. Okay. And this is the DNA polymerase. So that's how DNA polymerase happen. We will discuss these things also more in more detail in your next week's course content, I think. So this is a only basic, and there are many more enzymes are involved for this DNA replication, and we will discuss it eventually. So now we will have the problem number 11. So once I will show you the problem number 11, I will give you a 5 minutes of break. Okay. And just all of you do not leave the class. I will not leave the class. I will just keep my camera and my microphone off for just 5 minutes, and I will show you the problem number 11 before going. All of you do not leave the class. Try to answer the problem number 11 in the chat box. Once I just after 2 or 3 minutes I will check the answer. Okay. But in between I will keep my camera and microphone off. So let me show you the next problem. Problem number 11, it says which of the following statement regarding RNA is false. Number A, rRNA acts as a catalyst to make peptide bond. Option B, the amino acid sequence of proteins depend on the base sequence of mRNA. Option C, RNA polymerase 3 catalyzes, synthesizes the mRNA. And option D, RNA polymerase 1, they synthesizes rRNA. So all of you try. I will just keep my camera and microphone off for 2 minutes. Do not leave the class, okay? But try to answer the problem number 11 in the chat box. After just 2 minutes I will rejoin. I will just check the answer for the problem number 11. Meanwhile, all of you just read the question carefully and try to answer it, okay? Do not leave the class. Just try to answer the problem number 11 in the chat box. Hello. So is my voice audible, and can you look, I mean, is my video is visible also? If everything is all right, just let me check once. Okay, thank you very much. So yeah, I have given you the time for problem number 11, and I can see there are few answers for it. Okay, so most frequently given answers are number B, and so let us check what is the correct answer. So the question was, which of the following statements regarding RNA is false? Okay, so you have to identify which of the following is false statement. So the false statement is number C, RNA polymerase 3 synthesizes mRNA. So this is not correct. Okay, now let us check one by one. The first option, it says that rRNA acts as a catalyst to make peptide bond. So it is actually correct, because when I will show you the exactly the what are the roles of every RNA, like for rRNA, mRNA and tRNA, you will see rRNA, they act to catalyze the proper peptide bonds in the translation process. Okay, the amino acid sequence in proteins depend on the base sequence in mRNA. It is also correct, because I have shown you in the, I think I don't remember, but I have shown you in I think question number, let me show you. I have shown you this question, like here, as I have mentioned that the DNA sequence, if the DNA sequence is like this, and if it is a mRNA sequence, 3 of the mRNA base pair sequence, they particularly act as a codon or act as a code for a particular amino acid. Okay, and that's how the protein sequence or protein synthesis it happens. So that option is also correct, because the amino acid sequence will depend on the base sequence of the mRNA. Option number C, the RNA polymerase 3 synthesizes mRNA, it is not correct, and the option D is also correct. Now I will show you the RNA polymerase 1 versus 2 versus 3. The RNA, we will discuss the role of each and every RNA in detail in next class. Okay, because that is in your week 6 course content. But today I will just discuss a little bit of it. So RNA polymerase, there are 3 type of RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. So the RNA polymerase 1, that only transcribes ribosomal RNA, okay, or rRNA, but not 5S rRNA. So RNA polymerase 1 synthesizes rRNA, it is correct. RNA polymerase 2, it is a central enzyme that catalyzes DNA directed mRNA synthesis during the transcription of the protein coding genes. And RNA polymerase 3 is transcribes small untranslated RNAs such as tRNA. So in a simple language, RNA polymerase 1 synthesizes rRNA, RNA polymerase 2 synthesizes the mRNA, and RNA polymerase 3, it synthesizes the tRNA. So RNA polymerase 1 transcripts rRNA genes. This resides in the nucleus. The transcribes pre-RNA at 200 copies contain the same 2 non-identical alpha-like subunit and insensitive to a alpha amanitin. RNA polymerase 2 transcripts mRNA, miRNA, snRNA and snORNA genes. So these are different kind of RNAs. We will discuss it in detail in later. They resides in nucleoplasm, transcribe around 25,000 genes, has 2 copies of the different alpha-like subunit and sensitive to 1 microgram per ml alpha amanitin. And RNA polymerase 3 transcribes tRNA and 5S rRNA genes. They resides in the nucleoplasm, transcribes 30 to 50 genes at variable copy numbers and contain the same 2 non-identical alpha-like subunits and sensitive to the 10 microgram per ml alpha amanitin. So this was the RNA polymerase 1 versus 2 versus 3. You don't need to memorize everything, but remember it, like RNA polymerase 1 synthesizes rRNA, RNA polymerase 2 synthesizes the mRNA and RNA polymerase 3 synthesizes the tRNA. Just remember this thing. Okay. Moving forward, problem number 12, which says that what is the main enzyme component of the Sanger sequencing? Option A, helicase, option B, DNA polymerase, option C, nuclease and option D, guidase. Identify which of the following is the main enzyme component of the Sanger sequencing. All of you try to answer this. After that, I will show you what is the correct answer, what is the Sanger sequencing, and we will discuss it in detail. What is the main enzyme component of Sanger sequencing? Is it helicase, DNA polymerase, nuclease or guidase? Let me check the chat box. Okay, it is number D, number B, number B. So I have answers for number B and number D here. Okay, let us check. So the correct answer is number B, DNA polymerase. DNA polymerase is the main enzyme component of the Sanger sequencing. So first, before starting the Sanger sequences, let us understand what is a D-deoxynucleotide. Okay. So what is a D-deoxynucleotide? So as you can see, this is a particular nucleotide. There is a thymine base, and we have this particular sugar moiety here, the deoxyribose sugar. There is a 5' and 3' end. So and here also it is the there is also another, it is bonded with another nucleotide, and there is another nucleotide, but in between this second and this third one there is the single hydrogen atom as 3' carbon of sugar cannot form the phosphate with next to the next unit. Okay, 3' hydrogen cannot form ester bond with next nucleotide, so chain termination occurs. So which means as I have mentioned that if in this 2' position there is a hydrogen, so it is called the deoxyribose sugar. Okay, deoxyribose sugar which is present in DNA does not have OH, it only has hydrogen at 2 position, but it has actually OH in the 3 position generally. But in D-deoxynucleotide, what happens is third position also contain only hydrogen, not hydroxyl or OH. If there is no hydroxyl present in the third position, this phosphodiester bond will not be happening. So what will happen then? There will be a chain termination. Means the chain elongation of this DNA will not happen, because the one nucleotide will not be able to make bond with another subsequent nucleotide. Okay. So these D-deoxynucleotides are used in the Sanger's method. So what is the Sanger's DNA sequencing method? Okay, they synthesize complementary DNA strand in vitro. In vitro means in your the laboratory setup. Okay, not inside the I mean the live body, but or in the glassware, or we can say it. So in each tube normal nitrogen bases are given, D-deoxy nitrogen bases are given. Okay, D-deoxy nitrogen bases means there will be adenine. Okay, adenine means there will be adenine, there will be ribose, D-deoxyribose sugar, and there will be your phosphate. Okay, DNA polymerase enzyme will be added, primer, buffers and salts will also added. So how it happens? Like there is a single strand DNA with unknown sequence. Let's just say this is a single strand DNA with unknown sequence which is in blue. Okay, and it serves as a template. Then we will have DNA polymerase, and we have this DATP, DCTP, and these are the D-deoxy happen, and radioactivity level primer is there. So there are 4 reaction mixture are prepared. First we have D-deoxy ATP, and then we have D-deoxy CTP, and D-deoxy TTP, and D-deoxy GTP. Okay, and this A, C, T and G, they actually are the different your adenine, cytosine, thymine and guanine. Okay, this is second step. Then what happens? There is a DNA synthesis happens and we have the gel electrophoresis, and we will see there is a this kind of your sequencing plate we will show. Now, it will be seen by the gel electrophoresis by auto radioactivity. There is a shorter fragments in here in the lower side and longer fragments will be there, and the new sequence it will be you can read the new sequence like this and reduce the sequence of template by only changing the only writing the complementary strands. So, if the sequence come from this electroplate, it is like GACT, GAA, GC, the original will be CTGA, CTT, GCG. Okay. We will have a example for the DNA sequencing by Sanger's method in later in today's class, so do not worry. So, just remember, in Sanger method D-deoxynucleotides are added and DNA polymerase is the enzyme which is used here, and the gel electrophoresis technique it will be followed. Okay. Now, problem number 13, it says that which of the following statement is false regarding the DNA? Your options are: number A, all of the A DNA, B DNA and Z DNA have right-handed helix. Option B, A DNA is more compact than B DNA. Option C, most common DNA is the B DNA. And option D, A DNA has 11 base pairs in a complete turn. So, in DNA there are different kind of strands are there, 3 of which are A DNA, B DNA and Z DNA, and they differ in their characteristic for various cases. And try to answer this question. After that, I will discuss about all of these 3 DNAs in detail. What is A DNA, what is B DNA and what is a Z DNA and what are their characteristics. First, try to answer it in the chat box. Which of the following is false regarding the DNA? All of A DNA, B DNA, Z DNA have right-handed helix. A DNA is more compact than B DNA. Most common DNA is B DNA. A DNA has 11 base pairs in complete turn. Try to answer it in the chat box, then we will discuss about all the 3 of the type of DNAs. Let me check the chat box now. Okay, there are few options, option A, option A, option A, option A. So, option A is the most frequently given answer. Let us check if it is correct or not. So, yeah, option A, which states that A, B DNA, B DNA and Z DNA have right-handed helix, it is false. So, what is the different types of DNA? So, if you can see here, the structure, different structural forms of DNAs are given. This is the A DNA, okay, which is in this form. In B DNA, you can see the spacing is much more, and in the Z DNA, there is little bit of different kind of spacing, okay. If you say it the different forms of DNA like this, and if you see from top, this is the A DNA, this is the B DNA, it is the Z DNA. So, what are the comparisons? So, what are the characteristics of the all forms of DNA? Now, A form of DNA, it forms the helical sense, it is the right-handed, means it is a right-handed helix for the A DNA. For B DNA, it is the right-handed helix also, and for the your Z DNA, it is the left-handed helix, okay. Now, what is the diameter? The diameter means this diameter. If you see from top, this is the diameter, and it is looks little bit circular. So, the diameters are, for A form, it is the 26 angstrom. for B it is the 20 Armstrong, and for Z it is the 18 Armstrong. The rise per turn of the helix, the rise per turn of the helix means this from this point to this point, okay. So for case of A DNA, it is 28 Armstrong from here to here, okay. From B DNA it is 36 Armstrong, and from for Z DNA it is 44 Armstrong, okay. Base pairs per helical turn, so in A form there are 11 base pairs, okay. In the B form there are 10 base pairs, and in the Z form there are 12 base pairs. Helix rise per base pair, the helix rise is 2.6 Armstrong for A form, B form it is 3.6, and from the Z form it is 3.7. Base tilt normal to the helix axis is 20 degree for A DNA, it is 6 degree for the B DNA, and it is 7 degree for the Z DNA. Glycosidal bond formation is anti for the A DNA, for B DNA it is also anti, for Z DNA anti for pyrimidine and syn for purine bases, okay. So these are different characteristics of the different kind of DNA, but remember these points more are important, like the helical type, it is if it is right handed or if it is left handed, okay. The diameter, the rise per turn, okay, base pair per helical turn, so these are more important, and glycosidic bond formation also, okay. So this was all about. So first question, first option it was incorrect, which all of you have written in chat box very correctly. A DNA is more compact than B DNA, it is correct because it has in small place it has much number of base pairs, okay. Most common DNA is B DNA, it is also correct. A DNA has 11 base pair in a complete turn, it is also correct, okay. So now I will move toward the next problem. So problem number 14, it says that which type of topoisomerase which generally relaxes DNA by removing negative supercoiling is type 1, type 2, type 3 or type 4. So which type of topoisomerase generally relaxes DNA by removing the negative supercoiling? Try to answer this question in the chat box. Which type of topoisomerase generally relaxes DNA by removing negative supercoiling? Sorry. Okay, let me check the chat box. Okay, type 3, A. So I think this is the option for it, okay, type 3. Okay, let me check which is the correct answer and then let us discuss it. So the correct answer is actually it is number A, type 1. Okay, let us check what is a topoisomerase. So again remember in last slide actually I was discussing the DNA replication, and where we said that the enzyme, that two strands of DNA when they are just start the unwinding. So what happens, there is a tension is created between the point of the unwinding, okay. So why, if you you can do it actually very easily if you take two ropes which are in a helical formation, okay, and if you try to just separate them, you will see there is a more tension is generated at the point of the separation, okay. And this topoisomerase is kind of the enzyme which actually relaxes by this DNA and they can they can remove the negative supercoiling. So, supercoiling of DNA topology A, right-handed supercoiling means negative supercoiling, okay, and this is underbinding. Left-handed supercoiling means the positive supercoiling. Number C, relaxed state has no bends. DNA must be constrained, plasmid DNA or by proteins. Unveiling the DNA at one position changes the superhelicity, and topology only defined for the continuous deformation, no strand breakage. Okay, you have to remember no strand breakage. The topology, this term and the topoisomerase all are related with the deformation or but not for the breakage of the strand. So, there are basically two types of topoisomerase which are most common, the type 1 and type 2. So, what is the role of type 1 and type 2? Topoisomerase type 1, the enzymes which cut one of the two strands of double stranded DNA, relax the strand and reannul the strand. Okay. And topoisomerase 2, the enzymes which cut both strands of the DNA helix simultaneously in order to manage DNA tangles and supercoils. Type 1 generates a single strand breaks, type 2 generates double strand breaks. This occurs in eukaryotes, this occurs in both eukaryotes and the prokaryotes. It does not require ATP hydrolysis, this requires ATP hydrolysis. There are three subclasses. The type 1 is topoisomerase, type B topo1B and type 1C topoisomerase. There are two subclasses, type 2S topoisomerase and type 2B topoisomerase. Okay. So, remember the topoisomerase which relaxes the DNA by removing negative supercoiling is type 1 topoisomerase. Now, moving toward the next problem, the Maxam-Gilbert method of DNA sequencing is dash method for the DNA sequencing. You have to fill in the blanks. Option A, enzymatic method. Option B, chain termination method. Option C, D-deoxy method. And option D, chemical method. So, among these four, you have to identify which is the correct term for the Maxam-Gilbert method of DNA sequencing. Try to answer it in the chat box. The Maxam-Gilbert method of DNA sequencing is dash method for DNA sequencing. Is it enzymatic? Is it chain termination method? Is it D-deoxy method, or is it chemical method? Sorry. Everybody should try to answer this question. The Maxam-Gilbert method of DNA sequencing is dash method for DNA sequencing. Is it enzymatic? Is it chain termination method? Is it D-deoxy method, or is it chemical method? Let me check the chat box now. Number D. So, number D is the most given answer. Okay, let us discuss what is the correct answer and what is the Maxam-Gilbert method. So, the correct answer is number D. Yes, it is the chemical method. So, Maxam-Gilbert's method is a chemical method. That means he used a chemical reagent to know the sequence of bases present in the DNA. On the other hand, Sanger's D-deoxy method is an enzymatic method. He used an enzyme to know the base sequence of DNA strand, and as I have mentioned earlier, the enzyme which is used in Sanger method is DNA polymerase, right? So, what is the differences between the Maxam-Gilbert sequencing versus Sanger sequencing? So, in Maxam Gilbert sequences, what happened? The method of DNA sequencing is based on the nucleotide specific partial chemical modification and subsequent DNA cleavage. The Sanger method, the process of selective incorporation of chain terminating deoxynucleotides by DNA polymerase during in vitro DNA replication. It is the first one, it is developed by Alan Maxam and Walter Gilbert in 1977 and 1980, and the Sanger sequencing, it is developed by Frederick Sanger and colleagues in 1977. We all know the Frederick Sanger, he also have developed the Sanger's method for the protein, the peptide sequencing. Okay, we have learned it in, I think, in the first class itself. This first Maxam Gilbert method, it is a chemical method for sequencing, but the Sanger sequencing, it is a chain termination method. The Maxam Gilbert uses large amount of hazardous chemical including radioactive material and hydrazine, but Sanger sequencing, they use less hazardous chemicals. Maxam Gilbert is highly sensitive and highly specific. The Sanger sequencing is less sensitive and less specific. So, Maxam Gilbert method, what happens? Maxam and Gilbert, they use chemical reagent which induce the specific cleavages, and from this, by seeing the cleavages and the strands are formed by the cleavage, we can deduce what is the sequence for the particular DNA. And in the Sanger sequencing, the deoxynucleotides are used. We have discussed deoxynucleotides, what are those in the last to last slide, and this is the in vitro DNA replication, which uses less hazardous chemical, while the first one, the Maxam Gilbert, uses much hazardous chemical. Maxam Gilbert is thus a chemical method for DNA sequencing, and Sanger is a enzymatic method. So, the question may come as like this: what is the enzymatic method for DNA sequencing? So, what will be the answer? The answer will be Sanger sequencing. So, remember the differences and the characteristic of these two type of DNA sequencing. Now, moving toward the next problem. Problem number 16, it says that the combination of chemical condition for the thymine and cytosine specific cleavage in Maxam Gilbert method are: option A, hydrazine piperidine; option B, DMS or dimethyl sulfide and piperidine; option C, DMS dilute H plus and piperidine; and option D, hydrazine aqueous NaCl and piperidine. So, which combination of chemicals should be used for the particularly thymine and cytosine specific cleavage in the Maxam Gilbert method? I have mentioned it is a chemical method where different sets of chemicals are used to specific cleavages, and you have to now identify which is the particular correct chemical combination for T and C specific cleavage. So, let me check the chat box now. Okay, so the options are given like this. Number C. So, we have only one option. Okay, let us check. So, the correct answer is number A, hydrazine and piperidine. So, in Maxam Gilbert, carefully remember this combination of the particular chemicals. Okay, dimethyl sulfate or DMS and piperidine alone will selectively cleave guanine nucleotides. Okay, so DMS and piperidine, they will cleave the guanine specifically. Okay, but dimethyl sulfate piperidine in formic acid will cleave both guanine and adenine. So, if we have dimethyl sulfate piperidine and dilute H plus, means the formic acid or any kind of acid, so it will cleave adenine, also it will cleave the guanine. Okay. Now, hydrazine and piperidine will cleave thymine and cytosine all together. So, thymine and cytosine will be cleaved by hydrazine and your piperidine. So, T plus C, it is cleaved by hydrazine and piperidine. But the hydrazine piperidine in 1.5 molar of NaCl will only cleave the cytosine nucleotide. So, hydrazine aqueous NaCl piperidine, it will cleave the cytosine nucleotide. So, as you can see, there are 4 type of reagent combination for the Maxam Gilbert. Hydrazine piperidine combination, they will cleave thymine cytosine and cytosine, both thymine and cytosine. Dimethyl sulfate or DMS and piperidine combination, they will cleave only guanine. DMS in presence of DMS piperidine combination in presence of dilute H plus will cleave adenine and guanine together, and hydrazine in aqueous HCl in piperidine, it will cleave only cytosine but not thymine. So, these 4 are very important. This is very important. You have I will suggest, it is a very important question for this course also and for this week also. Now, what are the different chemical combination for the particular cleavage in the Maxam Gilbert method? Okay. So, now I will move toward the next problem. Problem number 17 says that which of the following statements is true regarding the double stranded DNA when it is heated above its melting temperature? Option A, there is decrease in absorption of UV light at wavelength 260. Option B, there is an increase in absorption of UV light of wavelength of 260 nanometer. Option C, there is no change in the absorption of UV light of the wavelength of 260 nanometer. And option D, it does not absorb the UV light at all. Now, find out which of the following statement is true regarding double stranded DNA when it is heated above its melting temperature. Your options are: number A, there is decrease in absorption of UV light of wavelength 260. There is an increase of absorption UV light of the wavelength of 260. There is no change in absorption, it does not absorb UV light at all. The 4 options, identify which of the following is the correct one. Try to identify what is the correct option among these 4. Let me check the chat box, then we will discuss it. Okay, the options are there. So, number B, number B, number A, number A. So, I have 2 number B and 2 number A. Okay, let me check now what is the correct option. So the correct answer is number B. There is an increase in absorption of UV light of wavelength 260 nanometer. So this is the figure, if you look at carefully. So in the y axis what we have absorption of 260 nanometer light, okay, and in the x axis we have the temperature. And so for the double stranded DNA, okay, so this is the this is the graph for the double stranded DNA, and this is for the single strand DNA, and this is this is what it is the melting temperature. So what the question is, the question says that double stranded DNA when it is heated above its melting temperature. So as you can see, when it is heated above the melting temperature, this is the melting temperature. Now if you increase the temperature, what will happen? There is an increase in the absorption of the 260 nanometer. Okay, as the graph you can see, it is the increasing, the absorption is increasing, means option B is correct. Okay. So now moving toward the next problem. Okay, it is regarding your Maxam-Gilbert sequencing. So write down the DNA sequence of the template DNA strand from the following diagram obtained by Maxam-Gilbert method. Okay, options are number A 5 prime G A T G C A T T prime 3 prime, option B 5 prime C T A C G T A 3 prime, option 3 C 3 prime G A T G C A T 5 prime, and option D 3 prime C T A CGTA 5 prime. This is the Sanger. So what happens in Maxam-Gilbert, I have mentioned it, like we have a 4 combination of chemicals, and each combination is specific for some specific cleavages of the bases. So if after the cleavage we just run the gel electrophoresis, you will have this kind of template, and from this template you have to identify what is the correct sequence for the given template DNA. Okay. Now carefully remember, try to remember what was the chemical combination, and look at the electrophoresis plate very carefully, where are the points given, and then try to answer this question. Just try for a few times, after that I will show you how to do it. Okay. Let me check, and there is a answer for it. Okay, so there are few answers like number B, number B, number B. So number B is the most frequently given answer. Okay, let me check if it is correct or wrong. Very good. So correct answer is number B. So how to do it, I will show you once. Since we have, so you remember in the last question we have, I have said there is a reagent in Maxam-Gilbert which is A plus G specific, one is G specific only, one is C cleavage specific, and one is C plus T cleavage. So what happens, it goes from here to here. this is the 5 prime, this is the 3 prime. Okay, and if you check it here, you will see here the spot is in the C plus T and C also. So when there are spot in these two, means the spot is due to C or cytosine. But if the spot is only C plus T but not in the C, means the base will be only thymine, the spot is for thymine. Again here, if there is a spot in A plus G but not in G, means the spot is only for adenine or A. And if the spot is in both C plus T and C, the spot is for C. If the spot is for A plus G and also G, it will be for G. And the spot is for C plus T, it is for only T, and if the spot is A plus G, it is only for A. So if you read it from here to here, it will be CTA, CGTA, CTA, CGTA, 5 prime to 3 prime. Okay. So this is about the Maxam-Gilbert method. Similar, we have another template. So write down the DNA sequence of the template DNA strand from the following diagram obtained by Sanger's method. So your options are: number A 3 prime GCTA GCAA CT 5 prime, option B 3 prime CCAT CGTTGA 5 prime, option C 5 prime CCAT CGTTGA 3 prime, and option D 5 prime GGTA GCAA CT 3 prime. So as you can see, there is the plate is given here. So you have to identify the common the correct sequence from the Sanger. So remember, this method is only for Sanger method. Okay, last one is for Maxam-Gilbert, but this is for Sanger. Now you have you have to identify the correct one. So take 2 to 3 minutes, try to solve it. Try to identify it. It is Sanger method. Remember, last one it was Maxam-Gilbert, but this is for the Sanger method. Look at the plate very carefully. Remember, try to remember what I discussed about the Sanger method. Sorry. So let me check if there is any answer. Okay, number C. So number C is given answer. So let us check and let us discuss. Okay. Correct answer is number B, 5 prime CT A CG TA 3 prime. Okay. So why it is number 3, and I think number C was given by you, but it is the correct answer is number B. So let me show you. Okay, sorry, I am very sorry, it was for. So the correct answer is here is number A. Okay, now let me show you why it is number A. So see, in the Maxam-Gilbert, it was when you are seeing a particular point at any particular base place, it is as it is. You have to take it as it is. Okay, so like what I have said, if the spot is in G, it will be only for G. But in case of Sanger's method, if the spot is in G, it is due to the C. I mean, we get the complementary strand in the Sanger's method. Okay. So if you see what is the Sanger DNA sequencing, PCR with radioactive dideoxynucleotides happen. Larger fragments are here, okay, and shorter fragments are here. Okay. So this is the again it goes to your it goes from the 3 prime to 5 prime, from here to here. Okay. and if it goes from C prime 3 prime to 5 prime here, okay, let me just draw it, write it here. So it goes from 3 prime to 5 prime here. Okay, larger fragment it will be in the 5 prime and smaller will be in the 3 prime. Now you have to take complementary strand for each and every. So here we have first your dot is in C, means we will have G at first. Then the second is also in C, so again first is on G. The second is on in the C, means second will be also the G. Third one it is in the A, means third one will be the thymine. Fourth one is in the thymine, means it will be for adenine. Fifth one it is in C, means it is for G. Sixth one is in the G, means it is for C. Again we have dots in 2T, means we have 2A here. Then we have in G, means it is the C, and we have in A, means it is for 3T. So remember here in the Maxim Gilbert you have to take the base as it is, okay, you don't need to do the complementary, but here you have to remember what the base pairs with another base and you have to write the complementary of, okay. So here decoding the sequence of interest is input, if the in plate it is showing like T, C, G, A, T, G, T, C, A, G. In the input strand it will be A, G, C, T, T, C, A, G, T, C and 3 prime, okay. So deoxynucleotides are added to normal PCR mixture which includes regular nucleotide, because the addition of deoxynucleotides to an elongating DNA sequence causes premature termination of that sequence. Radio labeling of the deoxynucleotides allowed the researchers to visualize the last nucleotide in each PCR sequence, okay. So it was for the your Sanger sequencing. Now is it understood by everybody? So is there any problem for the Maxim Gilbert and Sanger sequencing? If there is any confusion, let me know right now. I will repeat the both of the sequences once more, how the Sanger sequencing happened, how the Maxim Gilbert sequencing happened, okay, because this is a little bit tricky, but if you practice I think it will be easier for you, okay. Now let me know if you have you understood how the Sanger and how the Maxim Gilbert sequencing happened, how if a gel electrophoresis plate is given to you, can you deduce the original sequence for the Sanger and Maxim Gilbert method? I want all of you to respond in the chat box if you have understood or not, okay. If you did not understand it, I will repeat it once more. It may be little bit tricky, it may be little bit confusing, that's why I am asking you, have you understood it or not, or do you want me to just repeat it once more? So is it all right, or should I repeat it? Just remember what is the difference between the Maxim Gilbert and what is the difference between the Sanger. In Maxim Gilbert, the electrophoresis plate is here and the spots are for the sequence also. So if you are asked for the sequence, the spots will be the sequence itself, okay. But in case of Sanger method, what will happen? The Sanger method, it goes like this from 3 and you have to take the complementary strand, okay. So I want all of you to respond if you have understood it or not, or should I repeat it? Yeah, I will repeat it, don't worry. So in Maxim Gilbert what happened I have mentioned. So here it is the, if you have to remember it, okay. In case of here we have the sequence like this, okay, this is the 5 prime here, okay, I have just one mistake, I will just repeat it, and it is 3 prime here, okay, and you have to take the sequence from this manner, from lower to higher, and if in Maxim Gilbert the spots are as it is, you have to take, okay. So if it is the spot is showing in C, you have to take C. If the spot is in C plus T but not in C, means the spot is for T, and so on. That's how you have to take every spot, okay, as it is. But what happens in case of Sanger's method, okay, I have just made a mistake here and I will just erase it. So slowly eraser, okay, I have made a mistake here once, okay. So in case of your Sanger's method, what happens? Okay, in Maxim Gilbert it is lower one is 5 prime, upper one is 3 prime. Okay, and you have to read it as it is from 5 prime to 3 prime, from this side to that side. But in the Sanger method everything is your complementary. Okay, you have to remember everything is complementary. Here also you read it, you have to read it from this to this. Okay, here it is the 5 prime, here it is the 3 prime. Now if you read it from as it is from this to this, what the sequence you will get? You will get the sequence like 5 prime, you will have C, C, A, T, means the option number C, right? C, C, A, T, and we have again C, and then we have G, and we have T, T, G, A, 3 prime. So this is what we get from this electrophoresis plate, right? The sequence we get. But remember it is Sanger method, and in the Sanger method the original template DNA is just the complementary of this. So if we do the complementary of this, it will be 3 prime, G, G, T, A, G, C, A, C, T, and 5 prime, which is your this option. Okay. So in Maxim Gilbert you have to read the electrophoresis plate as it is, but in Sanger method you have to read it in the complementary method. Okay, if you read it from 5 prime to 3 prime in this code, you have to just do the complementary in case of the Sanger's method. Okay, you just look at the options carefully and try to practice it once. While I will show, when I will show you the PPT, I will share this PPT with you. See the examples very carefully and read it once. If you do not understand it again, I will repeat it in the next class. Okay, do not worry, I will repeat it in the next class if you do not understand it. So if you have any problem, you can let me know in the email also. I will repeat you there also. Okay. So last question of today's session. Okay. Okay, so sorry, here I have just forget to add the. Okay. So what is the restriction? So this question was regarding a restriction enzyme, BAMHI. So BMHI, so which cleaves between the base pairs. So your options were A and T, G and G, A and C, and A and A. So I forget to just add this in the presentation manner. So the options are given here. So okay, I will just answer this. So what are the restriction enzymes? Restriction enzymes are the enzymes that cuts DNA at specific nucleotide sequences known as restriction sites, found in bacteria and have evolved to provide a defense mechanism against invading viruses. In bacteria, they selectively cut a foreign DNA in process called restriction. To cut the DNA, restriction enzymes make 2 insertions, each strand of DNA double helix. So the restriction endonucleases, they digest double strand DNA at specific sites. The recognition site is in palindrome. So what is a palindrome? Palindrome is like this. The word example, madam, I am Adam. If you read it from backward, it is the same. So the sequence which read the same from the forward or backward, it is called the palindrome sequence. Okay, and reads the same on both 5 prime to 3 prime. Recognition sites are different sizes and unique sites, cuts between the same nucleotides on each strand. Okay. So if you check here, you will see if it is the your 5 prime to 3 prime, the here from this side, if you read the 5 prime, A, G, C, T, 3 prime, and if you see, if you read it from this side, it is same 5 prime, A, G, C, T, 3 prime. So this is called the palindrome syndrome, and these palindrome recognition sites are for the various restriction enzymes. Now different restriction enzyme, they cleave at different in between different bases, like AIUI, it cleaves between ALUL, it cleaves between GC and CG. Okay. HAE III, it cleaves between GC CG. BMHI, it cleaves between GG. HIN3, it cleaves between AA and A, and E. coli, it actually cleaves between G and A. Okay. AIUI and HAI3, it produce blunt ends, and BMH HI, Hind HI and Ecori, they produce sticky ends. So, these are the restriction enzymes. So, what you need to remember from this restriction enzyme, what is a restriction enzyme and where each and every restriction enzyme cuts, where this enzyme cuts in which, between which base pair, this enzyme, these are the very common examples, and I would in my suggestion you memorize all of these 5. Okay, I think each and you will get I mean common from these 5 options. Okay, so this was the problem number 20. So this was all of for today's session. So today what we did discuss about yeah. So okay, Bhavna, have you a problem with this question? I mean the Sanger's method. If you do not understand, I will repeat it once more, no problem. But you can tell me if you have not understand the particular DNA sequencing. Again, I am asking if anybody has any problem regarding the method of the sequencing. You can just tell me right now. I will repeat it. Okay. And I will just share this PPT, all of this with you, and you can just read it, and I think you will understand once you read it by yourself. Again, I am asking, do I need to repeat the sequencing? So, if I have to repeat the sequence, you can just tell me right now. I will then repeat the DNA sequencing by Maxim Gilbert also, by the Sanger also. Or have you understood it? Okay, so I suppose you have understand it. Again, I am saying, if you have any doubt, any query after the class also, you can reach me. I am sharing my email ID. Okay, okay. I think everybody has understood it. Okay, once so this is my email ID. So if you have any doubt or any query regarding today's class or any topic which I discussed in today's class, but your query was not answered by me, feel free to contact me. Okay, feel free to send me an email. I will repeat it. This today's lecture video and also this PPT I will send it to NPTEL, and they will share with you in soon. I mean in tomorrow and day after tomorrow they will share it with you, and you can just check the PPT and this video, and I think it will be helpful for you. Okay, so all the best for the week 5. This was your week 5 course content. So all the best for the week 5 assignment solving, and so this was your week 4 live session where we discussed the week 5 course content. So next Tuesday we will discuss your week 6 course content in the week 5 live session. Every Tuesday till the your courses ends, we will have this kind of problem solving session, and you can ask me any question related to the previous weeks also through the email or in the NPTEL question forum. Okay, I will happy to answer these queries, and thank you for joining in today's class. I hope you have learned something from your course, and I was able to help you, and I will see you in the next Tuesday at the same time from 6 p.m. to 8 p.m. Okay, try to join on time at the beginning so that we can discuss much more, and thank you all for joining. I will close the session for today. Just note down my email ID. So if you have any query, any doubt, just drop me an email. I will happy to answer your query. Okay, so thank you all for joining. Let us meet in the next Tuesday in the next class. I will close the session for today.